Greetings from the Science Connect. Welcome to our first edition of our new audio e-learning exploration series, Listen and Learn. Today we will explore and unravel the mysteries of messenger RNA vaccines.
Recently, Catalin Carrico and Drew Weissman were in news for being awarded 2023 Nobel Prize in Medicine or Physiology for their critical discoveries that enabled the development of effective messenger RNA vaccines against COVID-19. Through their groundbreaking findings, which have fundamentally changed our understanding of how messenger RNA interacts with our immune system, the laureates contributed to the unprecedented rate of vaccine development during one of the greatest threats to human health in modern times. Do you know what are messenger RNA vaccines? Through our new audio e-learning series, we will try to elaborate it for you. To begin with, first of all, we will talk on what are conventional vaccines.
A conventional vaccine, often referred to as a traditional vaccine, is a type of vaccine that uses a weakened or inactivated form of the disease-causing microorganism, such as a virus or bacterium, to stimulate the immune system. These vaccines have been used for many decades and have been highly effective in preventing various infectious diseases. Conventional vaccines work by introducing a non-threatening version of the pathogen into the body. This allows the immune system to recognize and respond to the pathogen without causing the actual disease. The immune response results in the production of antibodies and immune memory, so that if the person is later exposed to the live, disease-causing pathogen, their immune system can quickly and effectively neutralize it. Examples of conventional vaccines include those for diseases like measles, mumps, rubella, typhoid, polio, and hepatitis, to name a few. These vaccines have played a crucial role in reducing the incidence of these diseases and have contributed to public health by preventing illness and even eradicating certain diseases in some regions.
Thanks to the progress in molecular biology in recent decades, that today scientists have developed a new type of vaccine that uses a molecule called messenger RNA rather than part of an actual bacteria or a virus.
Messenger RNA vaccines are a revolutionary type of vaccine that have garnered significant attention, especially in the context of the COVID-19 pandemic. mRNA vaccines represent a promising alternative to conventional vaccine approaches because of their high potency, capacity for rapid development and potential for low-cost manufacture and safe administration. A key feature of mRNA vaccines is that they do not use live or weakened forms of the pathogens they aim to protect against. This is a departure from many traditional vaccines, which use either inactivated or attenuated pathogens to stimulate an immune response. The absence of live pathogen in mRNA vaccines means there is no risk of causing the disease they are designed to prevent. These vaccines utilize a novel approach to trigger an immune response in the body and have demonstrated remarkable efficacy. Recent technological advances have paved way for multiple mRNA vaccine platforms against infectious diseases and several types of cancer with encouraging results. Messenger RNA, often abbreviated as mRNA, is a type of ribonucleic acid, RNA, that plays a fundamental role in the process of protein synthesis within living cells. It serves as a critical intermediary between the genetic instructions encoded in DNA and the actual synthesis of proteins, which are the workhorses of biological functions in organisms. mRNA is a single-stranded molecule made up of a chain of nucleotides. These nucleotides are composed of a sugar phosphate backbone with one of 4 nitrogenous bases, adenine, cytosine, C, guanine, G, and uracil, U. Unlike DNA, which uses thymine, T, instead of uracil, mRNA contains uracil in place of thymine.
In summary, messenger RNA is a crucial molecule in the central dogma of molecular biology. It acts as an intermediary, carrying the genetic instructions encoded in DNA from the cell nucleus to the ribosomes, where these instructions are translated into proteins. This process is fundamental to all living organisms and is essential for their growth, development, and overall functioning.
The first step in designing an mRNA vaccine is to identify the specific antigen from the pathogen that the vaccine should target. The antigen is typically a protein found on the surface of the pathogen, such as a spike protein in the case of the SARS-CoV-2 virus, responsible for COVID-19. The spike protein is an ideal target for COVID-19 mRNA vaccines because it is the part of the virus that allows it to enter human cells. Once the target antigen is identified, scientists generate the mRNA sequence that encodes this antigen. This is done through reverse engineering the genetic information of the antigen. In the case of the spike protein of SARS-CoV-2, the mRNA sequence is created to encode a modified version of the spike protein. The natural mRNA molecule is not very stable and can be quickly broken down by enzymes in the body. Therefore, the synthetic mRNA used in vaccines is modified to make it more stable. Additionally, modifications are made to enhance the immunogenicity of the antigen, making it more effective in stimulating the immune response. To protect the fragile mRNA from degradation and facilitate its entry into human cells, it is encapsulated within lipid nanoparticles. These lipid nanoparticles serve as tiny delivery vehicles that protect the mRNA and help it enter cells more efficiently.
Messenger RNA vaccines work by leveraging the body's own cellular machinery to stimulate an immune response against a specific pathogen. Here's a step-by-step explanation of how mRNA vaccines work.
Step 1. mRNA encoding the antigen.
The core of an mRNA vaccine is a synthetic strand of mRNA that contains the genetic instructions for producing a specific antigen from the pathogen. In the case of COVID-19, this antigen is typically a modified version of the spike protein found on the surface of the SARS-CoV-2 virus.
Step 2. Vaccine administration.
A person receives the mRNA vaccine through an injection, typically in the upper arm. The mRNA molecule is fragile and can be broken down easily, so it needs protection to reach its destination in the body.
Step 3. Delivery in lipid nanoparticles.
The synthetic mRNA is encapsulated in lipid nanoparticles, which act as tiny delivery vehicles. These nanoparticles protect the mRNA from degradation and help it enter cells more efficiently. This lipid nanoparticle delivery system is a critical component of mRNA vaccine technology.
Step 4. Cell entry and translation.
Once administered, the lipid nanoparticles containing the synthetic mRNA enter cells in the surrounding tissue at the injection site. Inside the cells, the mRNA is released from the nanoparticles and begins to interact with the cell's own machinery.
Step 5. Protein synthesis.
The cell reads the genetic instructions in the mRNA and uses them to synthesize the antigen specified by the mRNA. In the case of COVID-19 mRNA vaccines, the antigen is the spike protein.
Step 6. Antigen presentation.
The spike protein or other synthesized antigen is displayed on the surface of the cell. The immune system recognizes these displayed antigens as foreign invaders.
Step 7. Immune response activation.
The immune system recognizes the antigen as a potential threat, leading to the activation of both the innate and adaptive immune responses. This includes the production of antibodies and the activation of immune cells, such as T cells and B cells.
Step 8. Immunological memory.
Importantly, the immune system remembers the spike protein or antigen. If the individual is later exposed to the actual virus, in this case, SARS-CoV-2, the immune system can quickly recognize and neutralize it, preventing infection.
Step 9. Antibody production.
The immune system produces antibodies that are specific to the spike protein or the pathogen's antigen encoded by the mRNA. These antibodies are the body's defense mechanism against the virus.
Step 10. Protection against future infections.
As a result of the mRNA vaccine, the individual becomes immune to the pathogen, or at the very least, is much better equipped to fight off the infection if exposed. This immune response provides protection against future infections.
The key advantage of mRNA vaccines is their speed of development and adaptability. When new variants of a virus emerge, the mRNA sequence in the vaccine can be adjusted relatively quickly to match the updated pathogen. This is particularly valuable in the face of rapidly evolving viruses like SARS-CoV-2.
In conclusion, mRNA vaccines represent a groundbreaking advancement in the field of immunization. They utilize synthetic mRNA to instruct cells to produce a harmless piece of the target pathogen, leading to a robust and specific immune response. The success of mRNA vaccines in the battle against COVID-19 underscores their potential to transform how we prevent and combat infectious diseases.